All right, let's continue with chapter 18. We're on section 3 now. So, when we think of DNA and chromosomes and genes, we think of genes as coding for particular proteins, which they typically do, and that's kind of how we define a gene. But it's a relatively small proportion of the genome, as you can see, that is of this coding variety. That is, codes for an mRNA that's going to be used to make a protein. So essentially, a significant portion of the genome is DNA that we would say codes for RNAs that are not directly responsible for making proteins or providing the instructions for making a protein. These non-coding RNAs would come under the variety of tRNAs, for example. They're used in protein synthesis, but they don't provide the code for making the protein. Ribosomal RNAs, so they would qualify as non-coding RNAs. But here are some others that you may not have heard of. MicroRNAs, small interfering RNAs. These are ones that play a role in the regulation of gene expression. MicroRNAs are small pieces of RNA that are made from a larger piece of RNA. You can see it has this rather convoluted structure to it, and small portions of it are snipped out by this protein called dicer, and they form a complex with protein whose job is to degrade pieces of mRNA that are no longer needed. So here in the post-transcriptional phase, an example of regulation where we basically break down mRNAs that are no longer needed. Or they can block the ability of ribosomes to make use of that RNA. So microRNAs have that level of control. And many genes are regulated in this way. RNA interference, these small interfering RNAs, as we call them, play a similar role. Curiously enough, they're formed from double-stranded DNA, although you can see that these microRNAs as well, because when they form these, so there's essentially a bunch of bonds there, so that little portion at least is double-stranded DNA, as you can see, or RNA, so that's kind of curious. They interfere with gene expression. That is where there is DNA that they are complementary to, that is the base pairs match up, they can prevent it from being expressed. They're also involved with the breakdown of mRNAs. Part of their reason for being, and perhaps the reason they had evolved, the book points out, is that they can block viral DNA. In the next chapter, we'll talk about viruses and how viruses basically inject their DNA into a host cell, and these microRNAs can help block that viral DNA from taking over the cell, essentially. So that's a benefit. They're also involved with modifying the shape of chromatin such that it either can, under certain circumstances, be transcribed and used, and other times not. And there are many other types of non-coding RNAs that you're welcome to look up. We won't talk about them here. Okay, so when genes are expressed, you can, or the modification of gene expression in time can influence the differentiation of cells and the development of organization. So to go from this fertilized egg, this zygote, to something you would recognize as some kind of organism, there's a lot going on, and there's a pattern of gene expression that regulates that process. And so essentially what these genes are, as we'll see, are doing is controlling the differentiation of cells. So when you have a fertilized egg, a zygote, you essentially have sort of the ultimate stem cell. It's an undifferentiated cell. It's just this zygote. Um, but over time, as it multiplies, as it goes through cell division, those cells start to become differentiated and specialized to become particular portions of the organism. And genes are also involved with not only the specialization of those cells, but this process of morphogenesis, the creation of form, the changing in the shape of that organism, as we said here, into something we can begin to recognize as a little tadpole. Okay, so first, cytoplasmic determinants. These are, oh, I got a typo there, organelles. These are um, compounds, structures inside of cells that can influence cell differentiation and development. So we see here, so we have our unfertilized eggs, the sperm cell, you get fertilization, you form the zygote, and even at the zygote stage, these little balls and triangles represent some hypothetical cytoplasmic determinants. And you can see they're not equally distributed. There's more of the balls on this side and the triangles on the right side. And when that cell splits in two, you've got more of one type on either side, and that begins the process of determining which developmental pathway those cells and the cells that they give rise to are going to develop into. So cytoplasmic determinants, and also this process of induction where cells communicate through chemical signals um, and can influence the developmental process as well. So basically internal compounds and signals and external signals can influence the developmental process. So we take a look here. So we've got this relatively undifferentiated cell, and it has a particular gene that's being expressed. And the expression of that gene causes the production of a particular protein, which leads to the determination that that is going to become a different kind of, a particular kind of cell. And that presence of that gene being turned on and that compound then begins a series of events that then leads to differentiation. So essentially you get first determination to become a particular kind of cell, and then it actually becomes that particular kind of cell. In this case, it's a muscle fiber cell. And generally, and particularly in animals, once this process of determination differentiation has happened, those cells, they can't go back to becoming some kind of undifferentiated cell. Once they've differentiated, that's kind of it. All right. So genes and their impact on the developmental process also leads to what's called pattern formation, ensuring that, as I say here, all the parts are in their proper place. Cytoplasmic determinants and induction are very important in determining this. So the pattern of an organism, like a fly. A fly has a backside, a belly side or ventral side, a head end, a back end, a right and left side. Excuse me. And so there are many genes that are involved in determining that the organism assumes its proper form. And these cytoplasmic determinants provide what you would think of as positional information. Essentially, the cells, by communicating with each other, kind of know where they are in the developing embryo, and they know what they're supposed to become. because of their position in the embryo. It's not talked about here. It's later in the book, but there were these bizarre experiments that people did where you had this embryo, and of course it consisted of a bunch of different cells, and so these cells had become differentiated. What they would do is sometimes in a second embryo they would take a batch of cells here and kind of stick them off on this end, and by this time they had already determined they were going to become a leg or something. And so then on this embryo that develops, you've got this leg kind of sticking off in a bizarre spot where it wouldn't normally be, because those cells had become differentiated. Fruit flies, here's that fruit fly, is an organism that's been used. We talked about it in genetics, how they've been very important. Well, they've also been important with studying the effect of genetics on development, and their development has been studied quite extensively. You don't really have to memorize this by anymore, but this just talks about the steps. Of course, you've got an egg, which is inside the follicle. The follicle is a structure in the embryo that contains an egg. And that egg sort of grows in size. And then after fertilization, you essentially have the zygote, which then goes through embryonic development. And the segments of an organism are formed due to instructions from genes and positional information, cytoplasmic determinants, and induction. And they have to have a particular pattern to them. These genes that control development are of a type generally known as a homeotic genes that control the formation of the pattern formation of the organism. And so here's an example of this was not due to moving portions of cells around in an embryo, but due to a mutation of one of these homeotic genes, one of these developmental genes, that in this poor fruit fly, instead of having where the antenna should be, it has an extra pair of legs, these rather misshapen legs. And so this rather monstrous mutation, again, due to some of these developmental genes, excuse me, that are not providing the proper instructions. Fruit flies in particular have about 13,700 genes, as we can see. We have more than that, but that's still a fair number of genes. And it's been estimated that about 1,200 of them are involved with development of one sort or another. Here's one of these that has been studied extensively. What's called bicoid. And it's a gene that is essentially codes for certain cytoplasmic determinants. It's what's known as a maternal effect gene. And so these, and by maternal effect we mean that the cytoplasmic determinants, remember the egg of course comes from the female, and the cytoplasmic contents of the zygote essentially come from the female, and so the mother has an effect on the distribution of these cytoplasmic determinants and essentially determines which genes will be expressed in which portion of the embryo. And so bicoid is an example of this. It is active in cells that are at the anterior end of the developing embryo. And so here, I'm not sure exactly the technique here, but they're somehow staining these and making these, they're tagging these active bicoid genes, and you can see they're clustered at this anterior end. And so it's a gene that is going to basically determine this is the anterior or head end of this embryo. And if somehow you were to transfer some of these cytoplasmic determinants to the other end, you'd have this monstrous creation. Okay, I'm going to have to finish the chapter in another video.